In this video, I'm going to talk about respiration, which is the release of energy from food. There's two main types of respiration, aerobic and anaerobic. Aerobic respiration is the controlled release of energy from food with the use of oxygen. And the reaction for that is glucose combines with oxygen to make carbon dioxide and water. Energy is a byproduct of this reaction and comes trapped inside molecules called ATP, which stands for adenosine triphosphate. We'll be dealing with this molecule later in this video. And anaerobic respiration doesn't require oxygen, unlike aerobic respiration, and it's also called fermentation. The first reaction here, where glucose gets converted to ethanol and carbon dioxide, is used in biotechnology to create alcohol. So biotechnology is the use of living things to manufacture useful products or to carry out useful reactions. And the second reaction is where glucose gets converted to lactic acid. This is what happens when you get a cramp. So if you're exercising and you get a cramp, that means that your muscle wasn't getting enough oxygen, so it produced lactic acid. Instead of carrying on with aerobic respiration and forming CO2 and water, it went with lactic acid. Now that we've talked about respiration, let's talk about energy carriers, which are molecules that contain energy entrapped within their bonds, and this energy can later be harvested or converted to other forms. ADP and ATP are important energy carriers. The structure of ATP is as follows. It has an adenine joined to ribose, which is joined to 3 phosphate groups. Now the name adenosine triphosphate comes from the fact that a bond, the joining of adenine and ribose makes a new molecule called adenosine. So when adenine joins to ribose, this whole molecule together is called adenosine. And it's adenosine triphosphate because you have 3 phosphate groups attached. The bond between the second and the third phosphate group has a high amount of energy, and when you exercise, this bond breaks and this energy is released as free energy. So all the energy that was trapped in that bond over there gets released as free energy, and you're left with ADP, which is a low energy molecule, and an inorganic phosphate. NAD+ and NADH are also important energy carriers. They carry electrons, which can contain high energy. NAD+ would join to 2 electrons and a proton to make NADH. And the energy from NADH is extracted using the electron transport chain, which we'll be talking about later. The next thing we're going to talk about is stage 1 of aerobic respiration, which is glycolysis. Glycolysis is the process where glucose gets converted to 2 pyruvic acid molecules. It's an anaerobic process, so it doesn't require oxygen, and this takes place in the cytosol of the cell. The cytosol of the cell is basically the cytoplasm of the cell without all the organelles, so the liquid part of the cell is the cytosol. How this occurs is that you have a glucose molecule. So this is the structure of glucose. It has 6 carbons. C6H12O6. The glucose molecule splits into two smaller molecules with 3 carbons each, called pyruvic acid. So this is the diagram of that, and this is just the same thing written down. So I've written 2 pyruvic acids instead of making 2 arrows over here. When this happens, you also make ATP. So 2 ADPs that were floating around in the cytosol join up with 2 inorganic phosphate groups and make 2 ATP molecules. And you also make water along with this. And this is called phosphorylation. Phosphorylation is when ADP joins with an inorganic phosphate to make ATP and a water molecule. You also have reduction going on in glycolysis, which is where NAD+ uses 2 electrons and a H+ from the glucose to make NADH. So the 2 electrons and a proton come from the structure of glucose. They get broken apart and join up with NAD+ and make NADH. And that's what makes 2 pyruvic acids. And the byproducts of glycolysis are 1 NADH, so there's 1 NADH there, and 2 ATP net. So there's 2 ATP produced each time you make 2 pyruvic acids. This is the actual structure, this is the actual reaction mechanism of glycolysis, which you don't need to know for the leaving cert, but I've put this in to show why I said 2 ATP net. I said 2 ATP net because you actually make 4 ATP, but then you use 2 ATP. So in the grand total, you've made 4 and you've used 2, so you make 2 ATP. So that's why earlier over here I said 2 ATP net. Next, we're going to talk about the Krebs cycle, which is a series of reactions in which a large amount of energy is formed, and this takes place in the matrix of the mitochondria, and this can only take place in aerobic conditions, unlike glycolysis, which took place in the cytosol and could take place in aerobic or anaerobic conditions. For the Krebs cycle, you have pyruvic acid, which was when you got what you got at the end of glycolysis. So in glycolysis, you broke a 6-carbon molecule, glucose, into 2 pyruvic acid molecules. Now, the pyruvic acid molecules, which has 3 carbons, is broken down into a 2-carbon molecule, acetyl-CoA, and a 1-carbon molecule, carbon dioxide. You also have the conversion of NAD+ to NADH when this happens. So again, the NAD+ gets its 2 electrons and H+ from the pyruvic acid molecule, so it breaks it apart from that, joins it to NAD+, and when you have NAD+, 2 electrons and a proton, you make NADH. So when you break pyruvic acid, you make NADH and carbon dioxide. Now from here on, the acetyl-CoA enters the Krebs cycle. And this is what the Krebs cycle looks like. The acetyl-CoA, which is a 2-carbon molecule, joins up with a 4-carbon molecule coming in. So there's a 4-carbon molecule, which you don't need to know about, but it's called oxaloacetate. It joins up, oxaloacetate, or the 4-carbon molecule, joins up with acetyl-CoA to make a 6-carbon molecule, which is citric acid, which is why this is also called the citric acid cycle. So the 4-carbon molecule joins up with acetyl-CoA, which is a 2-carbon molecule, to make a 6-carbon molecule. And then the 6-carbon molecule breaks and loses a carbon dioxide to make a 5-carbon molecule. And the 5-carbon molecule breaks and loses CO2 to make a 4-carbon molecule. And then you repeat, because another acetyl-CoA would be coming in, and this 4-carbon molecule would join up with that again, and then that would leave, and that would go through the Krebs cycle to lose another carbon dioxide over here, and another carbon dioxide over here. So here's some more reduction that's going on. In the Krebs cycle, you make 3 NADH molecules. So the NAD plus molecule over here would get its 2 electrons and hydrogen from here, from the 6-carbon molecule. This one would get its 2 electrons and hydrogen from the 5-carbon molecule, and this one would get it from the 4-carbon molecule. So that's the equation for reduction. You need to attach 2 electrons and a hydrogen or a proton to NAD plus to make NADH. In the Krebs cycle, phosphorylation also happens. So when the 5-carbon molecule is being broken apart to make the 4-carbon molecule, ADP gets converted to ATP. So ADP joins up with a phosphate group to make ATP and water. So that's the equation for phosphorylation. So these are all the products we've formed as from the Krebs cycle. So you've made 1 ATP molecule, which is over there. You've made 2 CO2 molecules, this one coming off and this one coming off. And you've made 3 NADH molecules, which is over here. And now we'll talk about the electron transport chain. Each NADH molecule is made up of 2 high-energy electrons, a proton, and an NAD plus molecule, so nicotinamide adenine dinucleotide. And the energy from these electrons gets used to make ATP. So these high-energy electrons, the high energy from them is going to be harvested, and it's going to be used to make ATP. And this process takes place in the inner membrane of the mitochondria, and it's an aerobic process because it requires oxygen. So NADH contains 2 high energy electrons, a proton, and NAD+. Now these 2 electrons are high energy electrons, so they have high energy, and in the electron transport chain we're going to have to extract this high energy from the electrons, and this is done in the inner mitochondrial membrane in some of the proteins, which you don't need to know about for the leaving cert. You need to know that they exist, but you don't need to know what they're called or go into any sort of detail in them for them. So these 2 electrons are going to pass through these proteins, and there's many proteins in the inner mitochondrial membrane. There's complex 1, 2, 3, 4, coenzyme Q, cytochrome C, but you don't need to know them for the leaving cert, but they're there, and the electrons pass through these proteins, and as they pass through, they deposit some of their energy into the protein and then pass on to the next protein, where they deposit some more energy and pass on to the next one, and eventually get down here to a low energy state. So when they get down to here, they have low energy. But as they were passing through these proteins, they were depositing some of their energy into these proteins, and this energy that they deposited is going to be used to make ATP. Now the production of ATP from these proteins is not on the course, so you don't need to worry about that. But you need to realize that as they go through the proteins, they're losing energy. And over here, it's a low energy molecule. From there, these two low energy electrons are going to combine with two protons and half an oxygen molecule to make H2O. So what's half an oxygen molecule? Well, an oxygen molecule is O2, which means that it's two oxygen atoms covalently bound together. So that's an oxygen, and that's a double bond between them. So that's what an oxygen molecule looks like. It's two atoms of oxygen covalently bound together. When you have half of O2, you just have one of these atoms. So that's what this over here means. It's just one atom of oxygen. So 2H is going to combine with an oxygen atom. So H2, which makes sense. You have two H atoms and an oxygen combining together, and you have two plus charges, and electrons are negative, so you have two minus charges. So everything balances, and you get H2O. So at the end of the electron transport chain, after all of this, they deposit all their energy into these proteins to make ATP, and at the end, just join up with H plus and O2 to make H2O.